Hello everyone, welcome back to another video from Shomu's Biology. In this lecture, we are going to talk about the cell signaling basics. Basically, this will be a series of lectures when we discuss about cell signaling processes. So the set of 3-4 lectures, you'll be understanding the basic principle of cell signaling. You'll also understand the components of a cell signaling pathway. You'll understand what is cell signal transduction, and you'll also see two examples of cell signaling pathway throughout this series. So watch all the videos from this series to have a better understanding of cell signaling. So at this moment we are going to discuss about the cell signaling basics. So basically, when we talk about cell signaling, what do we need to consider when we want to discuss about cell signaling? That's what we are discussing now. So I'll take this pen and paper. So just I'll take one color and move to explain. So let's say, okay, red color, fine. All right. So the very first thing that I want you to understand whenever you're talking about a cell signaling, I want you to understand one thing, that is the components of a cell signaling. Whatever kind of cell signaling pathway it is, whatever kind of cell signaling pathway, it is involved with some components. The components must be there in order to have a proper signaling approach. That's what we need to understand. So the first is cell signaling components. So what are the components that we're talking about? What do you mean by components? Basically, in the cell signaling process to occur, what is cell signaling in very simple idea? In very simple context, cell signaling is a process of cellular communication where one cell communicates with the other cell. Now maybe the two cells can be present in very close proximity, they may be present in very long distance, or they may be in contact, in physical contact to one another. In all these situations, still the cells need to convey message from one to the other, and that is done by the cell signaling. And the cell signaling is done with the help of what? Chemical components, okay, or direct contact. Even though they can involve in the direct contact, but still there are chemical components involved in the process of cell signaling. Okay, now what are the components of a cell signaling? Now before talking about the different components, let me give you a simple idea. Is that, let's imagine one thing, that we have this cell and it has an antenna like structure. So basically we are talking about an idea where two cells are talking to each other, right, conveying the message. So as we are conveying messages, what we do, we generally talk, right? Either you'll be in close proximity to me, so I'll talk directly to you, or you'll be someplace else, so I'll call you over phone, we'll talk. But ultimately we need a phone to talk, or a medium, that is words, to talk. Because if I say something, you'll understand, and based on that understanding, you'll respond. Same thing happens between two cells. Okay, this is, let's say, the cell number 1. So this is a signaling cell, and this is a cell who will receive the signal. So the signaling cell will produce something which will go and bind to the specific region of the recipient cell. So this is a recipient cell. So upon binding of this chemical molecules, remember the chemical component, upon binding of the chemical component to a specific region of the recipient cell in turn can produce something else which will even act on the signaling cell. So there will be a crosstalk between the two cells. This is known as crosstalk between the two cells. That means one cell sends something, the other cell will receive, and it will send something in return, the signaling cell will receive, and that way the process will complete. Okay, so for this simple process to occur, what we need a signaling cell, a recipient cell, but apart from that, we need a chemical compound, which is also known as messenger. So what do we mean by messenger? Messenger means it's a chemical compound which will convey a message from one cell to the other. And what else we need? We need a cell surface receptor. Cell surface receptor. So we need a messenger, we need a cell surface receptor. We need both. So here, upon binding of the signaling molecule or the messenger to the receptor, then sudden changes happen inside of the cell. This is known as intracellular changes. Intracellular changes inside of the cell. Okay, try to understand that. So with the help of this intracellular change inside of the cell, then ultimately there is a nucleus and specific instruction will come as mRNA to the cytosol, proteins will be made, and whenever we make proteins, we can make sure that those protein means expression. So again they can come out as a messenger and can relay the signal, may relay the signal, may not relay the signal. So this is an overall idea of cell signaling. So in this whole cell signaling part, there are individual components which play important role. What are those components? So let's see, these are the components. These are the components of cell signaling. Now if you want to remember this component part, you may think like how to remember all this. So for you I have a mnemonic, that is Firse. Those who don't know Hindi, it is again. Firse means again. So Firse is a term. If you remember Firse, you remember cell signaling. So cell signaling, Firse. So Firse, why? Because F is feedback mechanism, which is the end part. We'll discuss that. Then I is intracellular signaling. R is receptor. S is a signaling molecule. E is the effector proteins. So these are the components, right? So we need a signaling molecule, we need a receptor. These are the primary things that you need for a cell signaling. Without signaling molecule, without receptor, a cell signaling is not possible. So with the help of cell signaling molecule and receptor, cell signaling begins. So signaling molecule binds to the receptor, cell signaling initiates. Then that part is out. So that is the cell signaling molecules binding to the receptor is extracellular part, out of the cell, not inside of the cell. So what happens to inside of the cell? Inside of the cell, there is a response. The response that we see inside of the cell is known as intracellular response. And there are specific chemical molecules, proteins involved in the intracellular response. The intracellular proteins that are involved, they are known as, so either intracellular proteins or known as primarily they are known as secondary messengers. Why? Because the signaling molecule started the process of cell signaling. So the signaling molecule itself will be known as primary messenger. And the molecule which starts the process of cell signaling intracellularly will be known as secondary messenger. That's it. And at the end, when all this messenger signaling is done, the proteins will be made, because that is the expression. Ultimately, whatever cell signaling process is going on, whatever things are going on, the ultimate goal is to produce specific proteins in response to a particular signal. So whenever that specific protein is produced, that specific protein is known as effector proteins. Effector proteins, because they are going to show some effect. They are going to affect how the cell behaves, how the cell functions. Okay. Once the effector proteins are produced, those effector proteins can relay that message to the original initiating or signaling cell, that is known as a cell signaling crosstalk. And there is a feedback mechanism, which is very, very important mechanism, a feedback mechanism. This feedback mechanism either allows the signaling to continue or cause the signaling to stop. Because let's say we are considering production of a particular hormone. Let's say a particular hormone needed for that time period. So hormone is being produced by the regulatory cells, regulated cells or recipient cells. Signaling is done through brain, the hypothalamus and the part of that. So through brain it's being regulated. So whenever that hormone is produced in adequate amount, we don't need that hormone anymore, no longer need them. In that case, that hormone itself is going to shut off the brain center that signals the cell to release that hormone. That can be done. That is a negative feedback mechanism. So that's a mechanism of inhibiting a particular signaling. Similarly, in case of immunology, in immune system, what you find out is that T cells start releasing interleukins and they will bring more and more T cells. So whenever they produce a particular interleukins, they are going to bring more and more T cells, and they are influencing the number of T cell growth in the cell. That is the idea. So why that is? Because in that case, while they start growing, they are bringing more and more T cells to the place and start growing, start making more and more T cells, that particular part is a positive feedback loop. Because in that case, once a particular interleukin is produced, that interleukin is signaling the T cell to bring more and more T cells. That's a positive signal loop. But in case of hormone regulation, particularly in the regulation of ACTH, adrenocorticotropic hormone, thyroid stimulating hormone, the follicle stimulating hormone, for all these hormones, the negative feedback mechanism is always acting. Okay? So remember, फिर से cell signaling components clear. Alright, so now we'll move to the second part, which is related to the examples of all these different components of a cell signaling process. Remember, I told you in a cell signaling, there are all these different components. It starts with signaling molecule, you need it, without that it's not possible. You need receptor in the recipient cell, and there are intracellular signaling molecules and effector proteins, and last is the feedback mechanism, which is either positive or negative depending upon the situation where the cell signaling is going on. So at this moment what we are studying the components, right? Cell signaling molecule. So I'll tell you component one at a time here. So here, let's move on. Here are all the components listed, and I'm going to quickly tell you about all these different components. So here are different components listed, so I'll tell you about the role of different components that are placed here. Signaling molecule, it can be hormones, growth factors, neurotransmitters, cytokines, all of them. These are chemical factors, all of them can act as signaling molecule. They are acting as a signaling molecule. Receptor, two examples I'll tell you, and basically in the second and third, I think in the third and fourth lecture of this series of cell signaling videos, you will learn about individual receptor and their detailed pathway. One is G protein coupled receptor, also known as GPCR, and the second one is tyrosine kinase receptor, or RTK receptor, tyrosine kinase. Apart from that, there are ion channel receptors, intracellular receptors are also found, histidine kinase receptors are also found, but they are a little different. So these are the type of receptors that are placed there. Okay. Apart from this, what else we have? We have intracellular signaling molecule. Remember, apart from the signaling molecule and receptor, there are intracellular processes of signal transduction. So in the intracellular signaling, there are proteins, intracellular signaling proteins, also known as second messengers, because they started the process of signaling inside of the cell. So they are second messengers, because primary messengers are the signaling molecules. So second messengers example, cyclic AMP, IP3 DAG. IP3 DAG involved in the process of GPCR related pathway. We'll see that later. Protein kinases like MAP kinase, protein kinase A, protein kinase C. We'll see the role of protein kinases and MAP kinase in the receptor tyrosine kinase pathway. So because protein kinases are the second messengers of RTK receptor, and cyclic AMP, IP3 DAG, they are second messengers for mostly GPCR related pathways. Now the effector proteins are the ultimate transcription factors. They are going to cause specific transcription or transcription of specific genes, the product of which will move to the cytosol, and they will be translated into protein products, and those proteins are going to do their job, their effect. So they are the effector proteins. For example, transcription factors, CREB, CREB, NF-kappa B, ion channels, and several enzymes like adenylyl cyclase, phospholipase C, they all act as effector proteins. Many people confuse between the role of adenylyl cyclase and phosphorylase as a part of intracellular signaling protein, but no, they are not second messengers, they are actually effector proteins. Because effector proteins job comes after the role of second messengers. Remember that. So sequence goes like signaling molecules. Right after that, so signaling molecule binds to the receptor obviously. After that, the signaling begins inside. So second messenger. After second messenger, there will be effector proteins. Right? This is how the process works. Signaling molecule, second messenger, effector protein. This is the sequence. And cell signaling work in the sequence, and knowing the sequence is very, very important. So these are all the examples of different components of cell signaling. Now we are going to talk about the third aspect of cell signaling that you need to understand. These are the types of cell signaling that are present in a cell. What are the types? Remember, I told you that if we are standing in front of each other, that means we are in a very close proximity. So I can do two things. In very close proximity, I can touch you and ask something, or if I need to talk to you, I can simply tap and you look at me and I'll talk. That's one way. That is a direct contact. Remember, the whole process of cell signaling works by the same manner. Cell direct contact is possible. That's one kind of signaling. Second sort of signaling is that you're little, I can see you, but still I cannot physically touch you. So at that moment, I'll call you or wave at you. That is a signaling, right? Another way, that you are not in close proximity of mine, so I cannot see you, so I'll call you, right? So these are the three different types. Cell signaling is exactly similar in all these three different types. To remember these four different stages, I have another mnemonic for you, that is PE-DAS. Basically, to understand different aspects of cell signaling, I simply learned the name of two Bengali people. Das is the surname of Bengali, as well as Odia, they have DSH. But the thing is, Das, PE-DAS and Samir Das. These are the two names that I always say, Samir Das and PE-DAS. If you remember that, and you remember every single aspect of cell signaling, PE-DAS, Samir Das, फिर से remember this particular mnemonic, and you'll understand every single aspect. Why? Because in PE-DAS you can say PEDAS. Every single alphabet responds to some type of cell signaling. For example, P is for paracrine signaling. Paracrine signaling, P. Okay, so what is paracrine signaling? Paracrine signaling is when the cells are present in nearby proximity, but there is no direct contact. So that means you are standing somewhere, I am waving at you or calling you, and then the signal is going on. That is paracrine. Then E for endocrine signaling. Endocrine signaling works when, so E for endocrine signaling works in the long distance. Basically, let's say a brain cell is going to signal kidney. So that is a long distance. In that case, the signaling molecule need to pass through the bloodstream, and that is the signaling process. So there is a cell, and this is the bloodstream, and there is another cell. This is how it works. Okay, that is endocrine signaling. Hormones are examples of endocrine signaling pathway. They use endocrine signaling pathway. Alright, so endocrine is done. Then what we have? We have D. D for direct cell-cell interaction, direct cell-cell contact. So in this case, two cells will be in direct physical contact to each other, and generally they are connected via what is known as gap junctions. Via gap junctions. This is what they use for the signaling, direct cell-cell contact. Then we have A for autocrine signaling. Autocrine signaling is basically when a single cell is producing signaling molecule, then same signaling molecule is acting and interacting to the receptor of that same cell surface. So basically, auto-regulation or regulating itself is known as autocrine signaling, where the signaling molecule is produced by me. Let's say I am the cell, I produce the signaling molecule, I have the receptor, my signaling molecule binds to the receptor, and then the response is done inside me. That is autocrine signaling. And last one is S. S is nothing but synaptic signaling. This is where the neurotransmitters are the chemical messengers that are secreted by the nerve cells. They are involved in the synaptic signaling. This is where neural junction is where this process works. Neuromuscular junction is another place where this processes work. Means basically nerve cell releases chemical components that will act on our muscle cells and can regulate the muscle contraction and relaxation. On the other hand, the same signaling molecule released by a nerve cell act on another nerve cell. So from one terminal of the nerve to the another head of the nerve, and that way signaling process can continue. Both this type of signaling will be termed as a synaptic signaling, where they involve neurotransmitters as a signaling molecule. So I believe you know all this PE-DAS. If you remember all the different types of signaling process. You can take a screenshot of this particular slide and to get a better view of it. and can get to know about it more. So let's move on to the next part. We'll conclude our understanding of cell signaling basics with this particular slide, where I'll tell you one example of individual type of this signaling, right? So we are discussing about cell signaling, and I should not conclude this lecture without telling you at least one example of individual component of a cell signaling. For that we have this, and I'm going to share this list with you. This is the list, okay? So this is the list, you can clearly see. So we are going to see one example of cell signaling, and this is, we are seeing GPCR based cell signaling. GPCR based cell signaling. In the GPCR based cell signaling, again, like every signaling process, starts with cell signaling molecules. The cell signaling molecule, let's say in this case hormones. Let's discuss about the endocrine signaling. So adrenaline, glucagon, or neurotransmitters like dopamine, serotonin, they act as a signaling molecule in this process. The second thing are receptor. What kind of receptor? It utilizes beta adrenergic receptor, or if the signaling molecule is dopamine, then dopamine receptor. If the signaling molecule is serotonin, then serotonin receptor. Third one, intracellular signaling proteins. So after the signaling molecule binds to the receptor, extracellular part is done. What about the intracellular part? So obviously second messenger should be there, and also intracellular signaling protein in this case is G protein. G protein is trimeric. Three different units are there: alpha, beta and gamma. Alpha is the bigger unit, beta and gamma is little smaller unit. Now what is the role here is that whenever the signaling molecule binds to the receptor, signaling starts in the inside of the cell, intracellular signaling, the alpha unit separates from the beta gamma subunit, and they in turn is going to initiate the process and cascade of phosphorylation, which we'll see in the third and fourth lecture of this series. Then after that comes effector proteins. The effector proteins in this case can be adenylyl cyclase. Okay, if the pathway is cyclic AMP pathway, then the effector protein is adenylyl cyclase. If you are looking at IP3-DAG pathway, then the effector protein is phospholipase C. If you are looking at other example like ion channels, can also act as a effector proteins in this case. And finally, the feedback mechanism. What is the feedback mechanism used here? See, the feedback mechanism used is IP3-DAG is a feedback mechanism for the calcium. So calcium is used as a feedback mechanism, IP3-DAG act as a feedback mechanism. So basically in this case feedback mechanism stated this IP3-DAG pathway related to the idea that calcium which is produced in the cell and stored in the endoplasmic reticulum, the moment this pathway continues, so the calcium start to be transferred from the endoplasmic reticulum to the cytosol of the cell, and that act as a influx of calcium inside of the cell cytosol, and as a result of calcium influx, the further production of calcium will be inhibited. So that is the kind of signal that we are talking about at this moment. So this is one just example of this process. So I'll give you a simple idea. So here comes the cell and here is the receptor. Let's say the receptor is G protein coupled receptor GPCR. Signaling molecule, you will take any signaling molecule, the signaling molecule binds here. Let's say this is growth hormone that binds to the GPCR, or maybe any other kind of hormone that is linked with this pathway. Upon binding to the GPCR, then there is this internal cell, the effector, the intracellular signaling protein. The intracellular signaling protein is this is the G protein because they are related to GTP, and the moment the G protein means it is active when it's associated with GTP. Once the GTP is hydrolyzed to GDP, it gets inactivated. So it has an alpha subunit. Alpha subunit of this protein is released from beta and gamma. This alpha subunit is then going to activate adenylyl cyclase, which is an effector protein. Adenylyl cyclase will convert AMP to cyclic AMP. The job of adenylyl cyclase is to convert AMP to cyclic AMP. Once cyclic AMP is formed, then cyclic AMP will further activate proteins, which will further going to ultimately go and activate transcription factors. Transcription factors are going to produce specific mRNA and specific proteins will be produced. These proteins will then further do the job of and conclude the job of cell signaling pathway. So this is just an example, very basic simple example of cell signaling process in a cell. Okay, so this is the basic principle of cell signaling. If you understand this video well, then I recommend you to watch the later stages of this lecture, the other lectures that we are going to discuss. And also the next lecture is going to be on the topic signal transduction pathway. So if you want to know what is signal transduction pathway, what is the difference between signal transduction pathway term with the cell signaling term, then watch this particular series, watch the next video. And remember, in cell signaling you need to understand only two things, right? The earlier we discussed about Samir De and P they are फिर से. So Samir De P De फिर से. That's what you need to remember in order to understand all the different stages with a mnemonic. So that's it. If you understand and like this video, please hit the like button, share this video with your friends and colleagues, and subscribe to this channel to get more videos like that in future. Thank you. Bye.